Okay, so first we want to grab our crayfish from behind the claws so it can't pinch you, and then put it on ice to anesthetize it. We want to let it sit for about 10 minutes. Now that we've anesthetized our crayfish, we want to decapitate with the large scissors right behind the eyes. And now we can remove the claws. And then we'll want to separate the abdomen from the thorax. And we're going to switch to our smaller scissors.
Great.
So now we want to cut off our swimmerets to make our dissection a little bit easier. So now that we've separated the abdomen, we want to expose the muscles that we're going to be stimulating, which are located back here. And we also want to stimulate the nerve, which comes from the ventral nerve cord and extends out and innervates the muscles. So in order to do this, we want to stretch the abdomen out, and if you look, we want to cut along. There's kind of a dotted line that you can see that's formed by the joining of the ribs, and make sure to point your scissors towards the ribs so that we don't accidentally cut any muscle fibers. So then we just cut along this dotted line. And then we do the other side as well. And make sure you're pointing your scissors towards the ribs. So now we want to flip over the crayfish abdomen so that we can separate these top muscles from the muscles that we're going to be stimulating, which are found underneath. And if you can see, there's kind of a gap in here in which you can visualize the GI tract. And we want to use our forceps to carefully pull away the top muscle. Now we want to grab the GI tract and remove it. So now we want to remove the top muscle by picking up our small scissors and cutting across the piece up here. Now we want to place our abdomen into a dish and pour some crayfish saline over it. Now we can pin down the tail so that we can clean up the muscles a little bit. And I'm going to place one in the tail and then one in the middle up towards the top. We want to pull away the big pieces of muscle, but be careful not to damage the nerves underneath.
Great.
You might need to use finer scissors to cut away the musculature from the cuticle, and just make sure you can visualize the nerves and try not to cut anything that you can't necessarily visualize.
I wonder if you could just push on the end muscle and just kind of push it away, making sure that there's a gap. Is that possible?
Where is my...
Where do you want that muscle? Great, right? Okay. Fantastic. Yeah, we'll cut that in.
If we go to a higher magnification, we can visualize the nerves at a higher power. So this is the segmental nerve, which comes down and innervates the superficial lateral extensor muscle, which is a tonic muscle. And then it also comes and innervates this longitudinal muscle and the circular muscles, which are phasic. And these are the muscles that we're going to be stimulating. We want to split our prep in half so that we can have two functional preps. And to do that, we're going to cut right down the midline. And I'm going to grab with my tweezers and then cut down the midline, being careful to stick right along it so we don't cut anything else. And if you just go right between the two end muscles, you can see a nice line so we don't damage anything else. Now to remove these halves, I'm just going to cut across right here. Now I'm going to take these finer insect pins to stretch out the musculature, and we're just going to stick it into the cuticle to stretch it out. And that's to prevent the muscle from twitching so much when we stimulate the neuromuscular junction. So we want to come in and cut this little nerve coming out from the M muscle so that it doesn't twitch so much when we start to stimulate with the electrode. So now we're going to take it over to stimulate the motor nerve with the microelectrode and see if we see the muscle twitch. Okay, so now over here we have our stimulating electrode and our recording electrode, and we just want to put some wax down here to hold it in place. Okay, so this is our head stage for our recording electrode, and this comes over here and is connected to the extracellular amplifier, which then goes through the power lab and gives input to our computer. This is the suction electrode, and you can see there's this silver wire inside, and what I've done is I've just pulled up so that the saline comes up over the silver wire, and as you can see up here, I've already done it. And then that way when we stimulate, it'll come down here and stimulate between the two wires so that the nerve fibers get stimulated. So I've started a little bit outside of the nerve so that as I pull up on the suction, I can move in a little closer and it will just pull the nerve right up into the electrode. So next we're going to use an electrode puller and use this glass and this puller machine to make the actual electrode puller.
Okay, that sounds good.
Once we get our glass in place, we will actually do the pulling apart. So now we want to break the glass back. Now we're going to use this microscope so we can fire polish the tip so it's just not quite so rough.
Perfect.
Now that the filament's in place, we're going to heat up the filament so that we can smooth down the tip, just kind of like this, so that there are no more rough edges. Okay, so now that we have a smooth edge for our electrode, we want to bend the electrode a little bit because right now it would come in at an angle like this, but we want it to be more at a perpendicular so we get better contact. So what we're going to do is we're going to put the glass over the filament and heat it up so that it'll bend a little. Now you can see that the tip is curved, and it's important that we bend the glass slowly so that it comes out just right.